It's a pleasure to be presenting at the British Atherosclerosis Society today. I'm going to cover off a number of areas in relation to genomics of blood pressure. I'm Professor Sir Mark CAULFIELD from the William Harvey Research Institute at Queen Mary University of London. Here are my disclosures. Blood pressure is very important in stroke and coronary disease and is estimated to cause approximately 10 million deaths worldwide in 2017. It's also estimated that by 2025, there'll be 1.5 billion people with hypertension worldwide. Control of blood pressure remains challenging in spite of many good therapies. From familial aggregation studies, approximately 30% of blood pressure variation is thought to be due to genes, with lifestyle actually accounting for the remainder, and a complex interaction which forms a complex trait between lifestyle and genes. Rare syndromic forms of blood pressure have been identified, and these do allow us to target therapy to the genetic mechanism of the disease. Progress with complex blood pressure phenotypes has revealed many loci of modest effect, which explains why early studies were challenged in detecting loci when they were based on family-based linkage or small numbers of genome-wide significance. So we're going to focus in initially on blood pressure as a complex trait. And I'll focus immediately on this paper, Genetic Analysis of Over 1 Million People, which identified 535 new loci associated with blood pressure and was published in Nature Genetics in 2018. As a foundation, this used a discovery cohort comprising 500,000 people from UK Biobank with 250,000 from the International Consortium of Blood Pressure Studies. We analysed 7 million variants, and this discovery resource revealed 325 potential loci, which were replicated in the second stage with the Million Veterans Programme and the Estonians, a total sample size of 1 million. And then we, of course, were looking simultaneously at the Manhattan plot and noticed that many figures, many signals had reached genome-wide significance in biobank alone. So we then looked at a single-stage design using a more stringent p-value of 5 × 10⁻⁹. And this revealed a further 210 novel loci, which had a same direction of effect towards replication in the Million Veterans and Estonians, but were standalone loci that came from biobank alone, and illustrate the value of the scale of this resource. This gave us the 535 loci, and here you can see the Manhattan plot where systolic and diastolic blood pressure and pulse pressure traits are seen, all of these causing an effect size of 1 millimeter or less systolic or 0.5 millimeter diastolic. So these isolate certain biological pathways. The TGF-beta pathway, multiple components of that pathway are highlighted, which is known to affect sodium handling in the kidney, ventricular remodeling, and TGF-beta levels correlate with hypertension. Continuing that theme, vascular endothelial growth factor induces proliferation, causes migration of vascular endothelial cells, and stimulates angiogenesis. And alongside that, FGF gene enhances that by regulating VEGF-A signaling. And the urotensin receptor is a powerful vasoconstrictor receptor, which might be a suitable therapeutic target. And the relaxin gene encodes a G protein receptor that functions in vasorelaxation and signals through PI3 kinase. Interestingly, in druggability analysis, all the major current antihypertensive classes correlate with a gene target for blood pressure. And we detected in this analysis that the solute carrier SLC5A1 represented a potential repurposing opportunity, with the gliflozin type of anti-diabetic drugs being potential candidates, causing both blood pressure lowering and reduced glucose. So this has also been discovered in mortality trials where they seem to influence through mediating blood pressure the outcome. Then we moved on to create a polygenic risk score. The first of those was looking at the correlation on the left-hand side between blood pressure in red, systolic blood pressure, and we noted that all aggregate loci revealed a 13 millimeter rise in systolic, and that increasing deciles of the genotype risk score were associated with an odds ratio of 3.5. We also correlated this in the right-hand panel with all-cause cardiovascular disease as an incident disorder, incident MI, and incident stroke. And finally, we've looked at this in the ASCOT study, contrasting this risk score and between the lowest and upper quintiles of blood pressure, and discovering that 1.74 odds ratio exists for risk of resistant hypertension using this risk score. So this is potentially of clinical utility. So then we also, the Million Veterans have published a paper looking at that study, and that revealed 208 loci with common variation, 53 with rare variants across multiple ancestries. 53 were rare exonic variants. For example, one was in phosphodiesterase 3A, associated with various blood pressure traits. And there was also one in NOX4 redox pathway associated with pulse pressure, which might be due to arterial stiffening. And then my colleague, Patricia Monroe, with whom I work at the William Harvey, has published this analysis in December 2020, discovering rare variants associated with blood pressure in 1.3 million individuals. These were all focused on variants with a minor allele frequency less than 5%, and discovered 106 new associations. This is in keeping with the fact that those were mainly exonic variations, and the exon tends to harbor less variants associated with common complex disorders. Interestingly, a number of these were associated with reasonably sizable, 8 times larger than common variant effect sizes, and at least one was associated with a syndromic form where blood pressure phenotypes have been noted. That's the Adams-Oliver syndrome. Moving to looking at somatic mutation, which is mutations within our tissues, focusing on the work here of my colleague Maurice Brown at the William Harvey and Queen Mary, who has been doing exome sequencing and made multiple publications in New England, Nature Genetics, and has identified in the zona fasciculata KCNJ5 mutations, and in the zona glomerulosa, where aldosterone is typically produced, mutations in ATPase pumps, calcium channels, and the catenin beta 1 gene. So the frequency of these suggests a pre-existing drive for these mutations, but also more recently and excitingly published in July 2021 in Nature Genetics, a double mutation of GNA11 and catenin B1, which switches on the lutein hormone human chorionic gonadotrophin receptor, and also CYP11B2, which makes aldosterone in tumors in pregnancy. So this is an important additional evidence for somatic change causing blood pressure. TopMed have sequenced and released over 53,000 genomes with 410 million variants, and that's allowed us to create a new imputation scaffold. I'll come back to that in a moment. Exomes are now also available from Biobank, and a MEDX RIV preprint and 63,123 hypertensives and their controls have identified 2 exome-wide significance signals. And whole genomes will be available later this year on Biobank UK. And Olink are processing 50,000 of the Biobank samples using their Aptima monoclonal system. And all of us will present whole genomes on 1 million people. And of course, there's a 100,000 genomes project. And future, there will be Our Future Health. This is some work we've been doing in Biobank UK using 100,000 genomes. And here what I'm showing you in orange is the variants in TopMed that are not seen in Genomics England. In brown, the ones that overlap between TopMed and Genomics England resources, and in the green, the unique ones in Genomics England. And then there's also the overlap with the yellow color, which is a haplotype reference consortium. So what you see here is we can go from 34.88 million to several million variants, and this will refine our imputation scaffold further. And with Genomics England, we are creating an ancestry-specific imputation methodology that will, from what we've seen in blood pressure, allow extraction of greater value with new signals, and particularly fine mapping of signals based on added value from whole genomes. So blood pressure as a rare disorder is important. There have been various syndromic forms. Using the platform of the 100,000 Genomes Project, we looked at a family with focal segmental glomerulosclerosis, a father who presented in his late 20s, had dialysis age 29, a transplant at 30, retransplanted. Father, brother, and uncle had also died in the same condition, and his daughter also had high blood pressure and protein loss, and he'd sadly transmitted the disease to her. But because she did really good blood pressure control, her kidneys are working well. She's worried for years, as is he on the left, she on the right, that the young lady in the middle, who was in her teens then, now about 19, would have the same problem. And she'd been going to a GP for blood tests, urinalysis, and blood pressure measurements. And we discovered in the gentleman on the left an inverted formin 2 mutation, and that had been transmitted to the lady on the right in an autosomal dominant manner, as you might expect from the family history. And the NHS were able to test quickly within the young lady in the middle for this mutation, and to do a marvelous thing, which was to discover she didn't have it, and therefore she can stop going to general practice every month. And this is one of the most important things you can do in modern medicine, which is to provide reassurance. We've obviously looked at another bunch of families, over 138 with different mutations, a renin mutation, a COL4A5 mutation that may cause Alport syndrome, and 6.5, which is brachioautorenal syndrome. But we've only got a discovery rate at the moment that's very low, so we'd welcome you coming and helping us find new things. Just to touch on pharmacogenetics for a moment, I'm going to just share with you some of our work, on others' work on this. Based on the notion advanced by Nelson in Nature Genetics in 2015, that genomically primed therapies are twice as likely to make it to the clinic, we've been doing target validation and priming therapies from our work using dietary nitrate in the form of beetroot, CNP mimetics, because NO synthase and the nitric oxide pathway is heavily highlighted. Adrian Hobbs from the William Harvey has published in JCI on mouse models using a CNP mimetic and demonstrating efficacy, but this is being solubilized with money from Apollo Therapeutics to provide a viable drug for therapeutic trials in humans. And with GSK, Maurice Brown is optimizing CAV1 therapies to target adrenal adenomas. In very elegant work, Pino Bianchi and colleagues from Milan have demonstrated that genotype-driven clinical trials can work for reducing AA genotype, which predicts rostafuroxin as optimal working in antihypertensive therapy. And there's an ongoing piece of work in Glasgow, across Scotland, led by Sandosh Padmanabhan, evaluating the impact of uromodulan genotype on loop diuretic response in hypertension. Finally, to highlight the role of pharmacogenomics, on 76,000 whole genomes, 99.5% of us possess at least one haplotype that corresponds with a clinical pharmacogenetics international consortium gene-drug pair that may cause inefficacy or harm. In fact, 25.4% of us possess 4 such PGX genes. So PGX so far in blood pressure has highlighted the role of beta blockers and thiazides and genetic variation, but so far that's not been hugely helpful. This underscores the role of PGX in general. So ladies and gentlemen, these are the discovery papers in the last 10 years or so. And what you can see is the increasing study of rare variants in red, the increasing use of a single stage design due to the fact that we're running out of replication resources in dark blue. And all of these add up to increasing number of variants identified for blood pressure. And just looking at this, you can see moving on the bottom axis from rare to common variation, you can see the impact of rare variants on blood pressure is more appreciable, with some variants having between 6 and over 16 millimeters influence. I do, however, think it's worth dwelling on multi-omics and single cell technologies, because what we've shown you is in itself all very well, but if all it does is liberate biology we can't actually access, then that isn't that helpful. I do think there will be a role for long read whole genomes in the future. These allow us to read through regions we often can't read so well in the DNA, such as the HLA region, and repetitive regions such as the cytochrome P450 type 2D6. And also long-read genomes can generate methylation marks, and so you get epigenetics too. Transcriptomes are also going to be important, as is proteomics, which we now see being measured in high abundance via mass spectrometry or aptamers using low abundance protein measurements. And metabolomics, integrating all of these with single cell technologies where we studied tissue recently explanted from patients, though challenging in cardiovascular disease, and apply artificial intelligence, as we've seen in the work from Michael Simons from Yale, we can actually make intriguing observations that bring us much closer to the biological relevance of our findings. Of course, we still have experimental models, and these help us too, but sometimes they poorly recapitulate disease. But there is no doubt that if we're not going to simply generate with all of our genome sequencing and biobank our future health, etc., and all of us, and the 100,000 Genomes Project and the succeeding genomic medicine service, a whole bunch of variants of uncertain significance, we need to do more. And that's a national functional genomic strategy. So ladies and gentlemen, to summarize, we've discovered over 1,300 loci for blood pressure traits. Polygenic risk scores predicted 13 millimeter rise in the over 50s and correlate with cardiovascular outcomes. Rare variants will be important and may help us subset patients with blood pressure into different phenotypic groupings. Multiomics will be even more vital because it brings us much closer to the biology. And that includes evaluating tissue-specific impacts using single-cell technologies. I'd like to thank all my colleagues who've worked with me, both senior and junior, and my group at the William Harvey Research Institute. I'd like to thank the 319 authors from 165 institutions in 21 countries, and the generous funders, Wellcome Trust, MRC, BHF, and NIHR. And I'd like to thank the Society for your attention today. Thank you so much for having me here.